All right. First one. So this was from last week.
Yeah.
But it's to make viable sperm. What has to happen? First one.
Okay. Now, one of the things we need to understand when we're making sperm is that they have to have the right temperature. We need to be 5 degrees Fahrenheit cooler than body temperature. And for that reason, we have our gonads, the testes, outside of the body. Got it? Where do we keep the gonads for girls? Inside the body. Okay? Because to make viable eggs, it needs to be body temperature. In order to make viable sperm, it needs to be cooler than body temperature. Cool? Okay. We had a discussion this morning in my 112 class where they were going like, well, why would the testes not be cool enough? Because sometimes that's the problem. I said, the answer is tidy whities. Okay? And somebody said, who wears those? I'm going like, I don't, but if you go to Walmart, there's a whole shelf of them. So somebody's wearing them because they're not going to make them if nobody wears them. It pulls a little too close to the body, making them a little too warm, and the sperm might be created with a non-functioning tail. See? Well, they're not going to get to where they need to go. Make sense? Okay.
All right. What is gametogenesis?
Gametogenesis. Okay.
Gametogenesis.
Well, what's genesis?
To create.
To create. And to create gametes. So let's slide over here real quick. All right. What are the gametes? The gametes are haploid, meaning they have 1/2 of the chromosomes of the rest of your body cells. Okay? That's what haploid means. Half the chromosomes. And for guys, that's the sperm with 23. And for girls, that's the egg or the ovum, also known, I might as well add that here, an oocyte. Okay? 23. Got it? Now, when the egg and the sperm come together, they create a zygote, and that's the one with the 46. We call that diploid, which means double set. It's a double set of this. Got it? 23 plus 23. 10 4? So that's why those terms are terms that we need to be aware of. Okay? All right. How do I make a gamete? I use what's called meiosis. Now, mitosis has one split. Got it? You basically, in prophase, prior to prophase, you double the chromosomes, and then when you cut it in half, both of the daughter cells have 46 and 46. And mitosis is what we use for growing up, for replacement, for healing. Does that make sense?
Mm-hmm.
Okay. Now, what about meiosis? Meiosis, I have to do what? Cut the 46 into 23. So, the first step, we double the chromosomes again, and we make 2 daughter cells. But that's 46 and 46. I don't want that. So I make a second split, meiosis 2, where we split the 46 into 23 and 23. Got it? To make the eggs, to make the sperm. Now, as we go through this, I'll show you the difference between the making of eggs and the making of sperm. I don't know if that's that question, or there may be another one. Is there one specifically on spermatogenesis and oogenesis?
Not that I see. Or yes, there is right here. Yep.
So it's coming?
Mm-hmm.
Okay. All right.
The next one is function of the epididymis.
Epididymis. Okay.
Yeah.
Let's go to the videotape here. All right. See, and that's why meiosis has 2 sets. The first, PMAT, and the second, PMAT. Got it? Okay. Because we have the 2 splits. All right, and here's kind of what they're showing you. You got one, you got to split it, and then we have to split it again. Okay? So it's 2 splits. 1 becomes 2, then 2 becomes 4. All right? And we call this first split meiosis 1, and the second split is meiosis 2. all right? And we call these first cells that we start with the, whatchamacallit, spermatocytes. And then the primary spermatocytes go into the first mitosis, and the secondary spermatocytes go into the second mitosis. All right? And then you end up with these things. Now, these things are sperm, but they don't have any tails. We call these spermatids. Tids have no tails. Okay? Then later on, they elongate and get a tail. All righty. What was the question? Oh, epididymis.
Yeah.
Okay. Let's make sure we understand where everything is located. Sperm are made in the testes, and then they are stored in the epididymis. That's this thing right here. And now, the epididymis is a series of tubes, and while the sperm are in those tubes, they become motile. They learn how to swim. Now, I make a mistake and go like this. That's not how the sperm swims. How does the sperm swim? That's right. Okay, like a propeller. All right. During ejaculation, we empty this up the vas deferens, around the back of the bladder, and it comes into the back of the prostate. All right? Well, the only thing that is made in the testes are the sperm. It's the prostate gland and this back here called the seminal vesicles that produce the semen, the liquid that carries the sperm, alrighty, on their journey. 70% of the seminal fluid is made here, 30% is made here in the prostate, okay? What's in seminal fluid? Well, it's liquid, so it can travel. It is also contains bicarbonate. It has to be basic because you're going to be going down an acidic runway called the urethra, because what else has been going down the urethra the rest of the day? Urine, okay? So acidic. And that acid would kill the sperm. But your sperm also need a snack to fuel their tails. So there's fructose in the semen, got it? To provide that. Plus some prostaglandins and other types of paracrines to make them swim faster and to tell the vagina to do reverse peristalsis and bring them in to the uterus. 10-4? Okay. So your epididymis, there it is on your plastic model, that yellow one right here. Okay? And see, there it is. Okay? The sperm are made here, the little seminiferous tubules, and that's another question, so we'll talk about that specifically. And then they are transferred up here, okay, into the epididymis. Cool beans. Does that cover it?
Oh. Next one is the blood testes barrier.
Okay, well, let's talk about that.
Yeah.
That's going to be right here. So, when we are talking about these things, these little guys are called the seminiferous tubules. This is the birthplace of the sperm. And see, there's the storage area, the epididymis. All right? Well, let's talk about how this looks. If we were to cut through those tubes, it would look like this. Got it? This being the lumen, and this being the edge. Okay? Well, guess what? Sperm are made here, and as they mature, they get emptied into the lumen. So that right there are the actual sperm and their tails. Got it? So they start here and move this way. Got it? So let's take a look. There should be a picture that shows that. If there isn't, shame on me. But it should be in there. Here. Okay. This is it. See? Lumen on the inside. That's the edge. So we start on the outside here. We start with these diploid cells, and then we go through meiosis 1, then meiosis 2, spermatids, then they become sperm that we recognize with the long elongated tail. Everybody with me? Okay. But here is the the blood testes barrier. Okay? Out here is your blood vessels. Well, who's traveling around in your blood vessels? B cells and T cells. What are they looking for? Antigens, things that are foreign to them. Well, what is not foreign to them? Anything that they saw during training. Remember, they get their training, most of their training, prior to puberty. Well, when do guys start making sperm? After puberty. We start with diploid cells. Well, all of these diploid cells, the B and T cells recognize because they're diploid like the rest of our cells. Guess what they've never seen? A haploid cell. Got it? But they would never have seen that to begin with. So, you see this line right here? And you see this line right here? That's the edge of a cell. That's the nucleus of that cell. And this is the nucleus of this cell. And then it's going to be over here. And what is happening in the cell or right in between the cells? The creation of the sperm. You see these little things here? Those are tight junctions. They are part of the barrier because nothing from in here can leak out. And nobody from out here, B and T cells, can come in. That's why the testes are a privileged site that we talked about in the defense mechanisms chapter. We don't want B and T cells to see these because they'll want to attack them. Got it? We keep them away. That's your blood testes barrier. We call these cells by 3 different names. The easiest name is the nurse cell. They're also known as Sertoli cells because that's the name of the guy who discovered it. And they're also known as sustenacular cells because they help to sustain and promote the growth of these gametes. Got it? But the most important thing is that these tight junctions allow nothing to come out, so there's nothing for your B and T cells to taste and get fired up about. 10-4? Now, where do girls grow their eggs? In the ovaries. Are the ovaries a privileged site? No, because you are born with all the eggs. So you already have eggs, like up to 500,000 in your 2 ovaries, before B and T cells go to school. So yes, they know what a haploid egg looks like. Got it? It's considered self. What's considered non-self in a girl? The baby. So the uterus is the privileged site. The uterus is where we don't want B and T cells to go in or anything in there to leak out. Because the baby is obviously non-self. Because it's half you and half hubby. Got it? So your B and T cells would never have seen any of your husband's cells. Does that make sense? So that's why it has to be a privileged site, or else, all the babies would be killed as they're growing, or all the sperm would be killed as they're growing. But that's what a privileged site is. It's like keeping away the attackers, the B and T cells. Cool beans?
All right. Role of Cowper's glands.
Okay. Let's go back to our lab picture here. All right. Cowper's gland is right here. Okay. Another name is called the bulbourethral gland. It looks like a little bulb. Okay, next to the urethra. Alrighty? And that's this little guy right here. There's several of them you'll see on the plastic models that we identified last time. The purpose of the bulbourethral gland or the Cowper's gland is to put out a fluid prior to ejaculation. Got it? That fluid comes out first. And what is that fluid made of? Again, base, bicarbonate. And why? So we can neutralize the acidity of the urethra. Okay? So that comes out prior, and then we get the ejaculation. So it's a double way to make sure that we don't let the sperm get killed. Okay? Does anybody know why we have to make, there's over 150 million sperm in each ejaculate, okay? But there's only one egg. Why do we need 150 million sperm plus each time?
A lot of them die.
A lot of them die. So let's take a look at this model. I'll bring it over here. Okay? Let's take a look at this model here. All right? Here are the sperm going up into the vagina. 10-4. Now, because the cervix, which is the neck to the uterus, that's the neck, because the cervix fits into the vagina, there's going to be these little cul-de-sacs here on the side, all the way around it basically. Sperm only swim forward. They can't swim backwards. So if sperm swim up here, they get stuck and they die. So you're going to lose some sperm right there. Only the sperm that are smart enough to go up the middle into the external os, OS, the external os of the cervix, can go any farther. Got it? Once they get into the uterine cavity, some get lost in the uterine cavity and they die. Others will find their way to go down one of the two fallopian tubes. Well, how many eggs do you ovulate each month? 1. So this is the month that this ovary ovulates an egg. Oh gosh, that model, the egg is broken off. Sorry, there was an egg here. Okay. Yeah, notice it's a little piece of glue. The egg is gone. Oops. Anyway, so only the sperm that made it this way have a chance. What happens to all the sperm that went down the wrong fallopian tube? They're going to die. Got it? So that's why it takes many, many, many sperm so that one will make it and touch the membrane of the egg. Got it? Get it fertilized. Now, once that egg is fertilized, it does travel by reverse peristalsis, and cilia moves it back until it implants in the endometrial lining of the uterus. Cool beans? And because we don't want to have any more sperm interfering, we create a mucus plug. Got it? So the sperm might be here, but it ain't going to go any farther. Does that make sense? Okay. And the Graafian follicle, which is the tertiary follicle, that has ovulated the egg, under the influence of LH, becomes the luteinizing hormone. It becomes the corpus luteum, which means the yellow body. Now, what does the yellow body produce?
Estrogen
and progesterone. Okay. Now, what do those two do?
Keep you from going through menstruation.
Right. It's a negative feedback that goes back to the hypothalamus to tell the hypothalamus to create gonadotropic, inhibiting hormone, to tell the anterior pituitary, don't release FSH and LH, which would cause you to lose that lining. You don't want to lose the lining because there's a baby implanted in that lining. Does that make sense? Okay. So that's the purpose of the corpus luteum. What if there's no baby? Then the corpus luteum dies and becomes the white corpus albicans, which we don't see here, but there is in the male and the female part. Okay? So that's how that works. Who tells the corpus luteum there's a baby?
The baby.
The baby. What's the baby make? We did this last night. HCG. Got it? Human chorionic gonadotropin. Who better to know there's a baby than the baby itself? Okay? Now, back in the old days, You would have to go to the doctor, have some blood drawn, and they would do a blood test to see if you have HCG in your blood. Got it? You know, you missed your period, and you're thinking you might be pregnant. That's how they would confirm it. Now you can go to Walgreens or to Walmart and get yourself an in-home pregnancy test and just pee on a stick, because it turns out HCG is in your urine. And then you can check to see if you have HCG, you're pregnant. No HCG, you're probably not pregnant. Does that make sense? Okay. What else do you need, my friends?
Now we're on to the spermiogenesis and spermatogenesis. That
Okay.
one.
Okay. Let's see if we got a picture here. We should. Okay. Oh, okay. So you're gonna be a troublesome maker. All right. Let's draw on the board. Let's draw on the board. It's gonna be a whole lot easier. Let me get rid of this. Yeah, the new book kind of takes a lot of shortcuts, unfortunately. I'm into long cuts, okay? Yes, much to the detriment of my students. Ready? Spermatogenesis, okay? So here's what we're going to talk about. We start with the primary spermatocyte. This is the one that's going to be turning into two secondary spermatocytes. Got it? Primary, secondary. Well, what's happening here? This is meiosis 1, the first split. Okay? Well, what's going to happen next? Meiosis 2, the second split. And that's going to make 4. So this one is the primary, this is the secondary spermatocyte, and then these guys are the spermatids. Spermatids. Spermatids have no tails. Got it? And then they will become the sperm that we all know and love. All right? Does that make sense? Okay. And how many sperm do we make? 4. We need to make as many as possible. Okay? Well, let's compare this, and I'll switch to pink. Let's turn to the female version. Am I still on the computer? If I go over here?
Mm-hmm.
Okay. And that would be oogenesis from oocyte, okay, the egg. Again, we got the primary, alrighty. We're going to go into meiosis 1, meio 1, to make the secondary. But here's the difference. I need to make a correction here. You see, my friends, how many eggs do we want? 1. So we have 1 viable egg and a little tiny bag holding half the chromosomes. Because that's the purpose of splitting it. You want to go from 46 to 23. Got it? So this is called the first polar body. And it's called polar because they kind of end up hanging around one pole of the egg. Okay? So 1 viable egg and a polar body. Alrighty? Well, don't we have to do meiosis 2? Yeah. Meio 2. And this splits 1 viable egg and a second polar body. Okay? Now, sometimes this first polar body can also split. So, what's the bottom line? Here we have one viable egg and 3 polar bodies. And what's the bottom line here? 4 viable sperm. Does that make sense? Okay. Now, just because they need to make it a little bit difficult, from going from spermatid to sperm, got it? That's the actual sperm. This process, just this step, got it? Just this step is called spermiogenesis. Spermiogenesis. Got it? Just this last step. Okay? The whole thing is called spermatogenesis, but they got to make it a little complicated. Going from tids to tails has its own name. Okay? It's just part of the greater thing. Now, what do we need to know here? We do not undergo meiosis II until fertilization happens. We don't kick out the second polar body until the sperm fertilizes this guy. Got it? Why? Because what's the purpose? If we're going to lose this in the menstrual flow, why go through the process of splitting out half the chromosomes? We only need to split out that other half of the chromosomes when a sperm is coming in. Got it? So we have a 23 and a 23 instead of a 23 and a 46. Got it? We got to split that. Got it? So then I can be fertilized. Got it? So the trigger to going into meiosis II, the trigger for making the second polar body is being fertilized. Cool beans? Okay. What else can I do for you?
All right. Role of sympathetic and parasympathetic nerves.
Okay. Basically, you have both sympathetic and parasympathetic nerves running down to the nether regions, okay? Your erection is based on parasympathetic nerves. Got it? So, rest and digest, 10-4, is responsible for that. And that's why I jokingly say on the video, if you're having difficulty, you don't want to yell and scream, "What's wrong with you?" and beat it against the headboard, because that's not going to help. Got it? Because that's more sympathetic. 10-4? All right. She goes, "Oh, okay, now I know what to do." All right. So yeah, just beating it against the headboard is not going to help. Okay? But is sympathetic involved? Yes. Parasympathetic is for the erection. Sympathetic is for the ejaculation. So they both work right there. And this is why guys, and I say that because I'm in that age, guys are worried about getting prostate cancer because when you do prostate surgery, you might cut through the sympathetic or the parasympathetic nerves while you're down there operating. And guess what you end up with? ED, okay? Erectile dysfunction. And it's something you can't do anything about because it's the nerves that are required to help that happen, okay? So, and even if you have radiation, depending on how much radiation they hit you with, it could damage those nerves as well. So that's why a lot of guys are going like, "Please don't let me have a bad, PSA test." Prostate-specific antigen. When your prostate, this is for the guys, when your prostate is in trouble and starts becoming cancerous, it releases another protein into your blood which you can test for prostate-specific antigen. Got it? And that's why there's a limit, If it's this number or lower, your prostate's probably in good shape. But that's why they want to do a prostate, besides the exam. but they want to do a prostate PSA test every year because they want to see is the numbers going up. Got it? If the number's like this, you got nothing to worry about. If it goes like this, we got a problem. Okay? All right. What else?
All right. I'm gonna mispronounce this. Function of the gubernaculum.
Gubernaculum. Okay.
Yeah.
All right. We laughed about this in my class the other day. Your gubernaculum sorry. Yeah. It's an unknown number. All right. I think I saw a picture of the gubernaculum. Yeah. I'll get to you. All right. Okay, here we go. Your gubernaculum is this ligament. It's anchored at the bottom of the scrotum and to the bottom of the testes in a fetus. Got it? As you get older, that gubernaculum gets shorter. What's it doing? Pulling the testes down into the scrotum. 10-4? So, when the testes is all the way descended, it's because the gubernaculum has gotten shorter. So I, again, jokingly say the gubernaculum pulls your goobers down. Okay? This morning, my students going, what's a goober? I said, it's another name for a peanut or a nut. Well, aren't we pulling the nuts down? That's right. And that's why we call them goobers, because that ligament was called the gubernaculum all along. Okay? So it pulls the goobers down. What would happen if they did not descend? What would happen if the testes did not descend?
Could make viable sperm.
Okay, number one, it's too hot to make viable sperm, so you'll be infertile. But the second thing, and worse, you would get testicular cancer. Because when the cells are not growing correctly, when they grow incorrectly, we call that cancer. So you could very easily be making cancer. So that's why the doctor will feel for this on a newborn baby boy and see if he can ease this down. If for some reason he can't, then you have to schedule a surgery and have it surgically put down, because it's got to stay, remember, 5 degrees cooler than body temperature to make viable sperm. Cool beans? All right, what else?
All right. What is crossing over and where does it occur?
Okay, the crossing over, let's see if it's in here. I thought I saw it in here. No, maybe not. If it's not, then it might be in the next chapter. Oh, here we go. Here's crossing over. Okay, here's crossing over. All right. See how we have 2 sets of chromosomes here? Let's assume the blue one came from mom and the green one came from dad. Got it? It was 1 and 1. Got it? You inherited 1 chromosome from your dad and 1 chromosome from your mom. Got it? But what happens when we have to get ready to split? We double the chromosomes. How do we double it? This becomes 2. Got it? And they're attached in the middle. So 1 becomes 2 with an attachment in the middle. Got it? All right. Now, here's the thing. In prophase 1, that's when this happens. Prophase 1, we end up putting all of the like chromosomes next to each other. So homologous chromosomes. Why am I saying this? Because this set of chromosomes, this might be for your ear, your eye, and your fingers. Got it? And same thing, ear, eye, fingers. Because we're going to switch some things here. Got it? That's what we call crossing over and then recombining. What used to be the chromatid here becomes the chromatid here, and vice versa. Now, why is that important? Because when we split these chromosomes up, this gamete will look like grandma. This gamete will look like grandpa, but this gamete will have both. And I say grandma and grandpa because this is what you inherited as the mom or the dad. Got it? And why does this matter? Because we want diversity in the next generation. Alrighty? We don't want to make a clone of ourselves. And so, where does diversity take place? It takes place in three parts. This is one of them, crossing over during prophase 1. What's the second place we have? In anaphase. Anaphase 1. When we have anaphase 1, we could line them up blue, green, and blue, green like this, or what if it was like this? You have blue, and green. Got it? And then they would split. Then this would be all green, that would be all blue. Got it? Anaphase is where we separate the chromosomes first. So anaphase 1. 10-4? And that will determine, again, how diverse the gametes will be. And what is the number one area where we have the most diversity? Fertilization. Because it's not your own egg and your own sperm. It's your egg, somebody else's sperm. Got it? Your sperm and somebody else's egg. That's the biggest combination of two different individuals. Got it? And that's why all animals and all plants and mushrooms on planet Earth do sexual reproduction. Why? Because higher order organisms need a way to change over time. We call that evolution. They need to evolve. Now, why do we want them to evolve? We want them to have bigger heads and be smarter. No, we need to change to fit, survival of the fittest, to fit a changing environment. And I often bring this up, I don't know if I did it in my video, but I often bring up the fact that you heard of the Ice Age, right? The ice from the North Pole went all the way over Canada, all over the north part of the United States, and stopped at the Mason-Dixon line, okay? So it was very cold in North and South Carolina. In fact, who lived here and survived the cold? A woolly mammoth. And we know that because we found their bones in the Congaree Swamp. Got it? So we know they lived here. And there's models that they've created and put into the State Museum. Well, how come when the ice went back, we don't have any more woolly mammoths? Because now it's too warm for them. Got it? But the reason they survived is because they had the genes for having longer hair. And the other elephants that lived here didn't have that. Now, there was no ice age in Africa. Got it? That's why the elephants there still have very short or almost no hair. Because they never needed to have a warm coat. Does that make sense? But that's why every organism wants built-in diversity. Got it? Now, how come bacteria and whatnot, 1 becomes 2, 2 becomes 4, all the same thing? Because there's so many of them. They can multiply so much faster than we can that they can change with the environment by sheer luck. Got it? They simply build up resistance against penicillin. So everybody who gets killed by penicillin gets killed, but the few that can survive take over. Got it? And then we hit them with another amoxicillin. Okay? Those that amoxicillin can kill die. But those who genetically have a mutation lets them survive. Got it? And now they're resistant to that. 10-4? And that's how resistance happens in those small organisms. They can build themselves over and over and over and over again quicker than we can. Got it? In one of our generations, they go through hundreds of generations. 10-4? But us higher order animals and plants live longer. Got it? So we reproduce in a slower pace, but we still have to have diversity built in. Sexual reproduction is the best way because it's two individuals that are different from each other coming together. Okay? All right, what else?
All right. Purpose of mitosis, purpose of meiosis. I feel like we
Pretty much
Pretty
done
much
that, yeah.
covered
Mitosis, we only split once to make a duplicate of the original. That's for growth, that's for healing, that's for replacement, repair and replacement. Meiosis, on the other hand, is only done by the gonads, only done by the ovaries in girls, only done by the testes in guys, and that's to cut the chromosome number in half. That's why we have not one split, but two.
real quick. Will you say, just as jumping back to the last question,
Yeah.
will you say again where crossing over occurs?
In prophase 1,
In prophase
during
one?
that step of meiosis, is prophase 1.
Okay.
Because in prophase 1, they all line up.
Okay. Yeah.
Yeah, and when they line up, they can switch.
Cool.
Okay.
All right. Next up, this is going into 28.
Okay, the female part?
Yeah.
Okay, let me pull up that.
Are we gonna do 20 and 21?
Yes, ma'am.
And 21?
If you want. All right. Lay it on me, brother.
When does the second polar body get ejected?
Okay, we know that.
Yeah.
Second polar body is not ejected until fertilization happens. Cool.
Next up, role of vestibular glands.
Okay. The vestibular gland, it's right about here. Okay. There's not on this model, but there is in the plastic models that we use. Of course, not on this plastic model. All right. The vestibular gland is analogous to the Cowper's gland. It's what makes the vagina lubricated. Okay? That's all there is to it.
All right. How does fertilization? How does fertilized egg move down the fallopian tube?
Okay, I can do that one right here. Assume there's an egg here, alrighty? Remember, it moves because of reverse peristalsis. We squeeze behind it to make it move towards the uterus so it can be implanted. Okay? I'll do it here. So, and cilia. There are cilia on the inside of that tube that move the egg, the fertilized egg, along its way. Okay?
What triggers menstruation? What is shed?
Okay. I think we have a good model of that. See? There's your little uterus and stuff. When there's no baby in it, it's about that big around. All right. Let's see. What was that again?
Let's see. What triggers menstruation? What is shed?
Oh, okay. Let's do that. All right. Before we go any farther, this is how we build up to an egg. Okay. We've already created it. Remember, you're born with all of them, and the primordial follicle under the FSH and LH gets turned into a primary, which goes into a secondary, which becomes a tertiary. We recognize the secondary because it has an antrum, a hollow space, okay? And the tertiary one, also known as a Graafian follicle, named after Dr. Graafian, is the one that gets ovulated, okay? Now, what causes ovulation to occur? A jump in LH and FSH, all righty? And then you ovulate. What's left of the follicle is not done for. It gets turned into the corpus luteum because of LH, luteinizing hormone. Got it? And this is the one that makes the estrogen and progesterone to feed back and say, don't do FSH and LH. There may be a baby on the way. If there's no baby, it dies. It becomes the corpus albicans, the corpus that is white. Okay? All right. And there you go. Show you some stuff. do we have what I'm looking for? This. Here's what we're looking for. I like this. All right? What do we have here? The events in the ovary with what your egg is doing, superimpose over the events in the uterus and what the uterine lining is doing. Okay? All right. So here we go. FSH and LH is going to make your egg mature. Got it? Then a surge of both, but mostly LH, is going to make you ovulate. At the same time, we just shed this lining. Okay? Now please note, you've got two layers here, the brown layer and the red layer. The brown layer is called the stratum functionalis. That's the one whose function is to let the baby implant. So when the baby is not there and we have a menstrual cycle, that's the layer you shed. Okay? The layer underneath, the stratum basale, you never shed, because that's the one that we grow the new one from. Okay. Cool beans? All right. So, what causes your menstrual cycle? FSH and LH. Got it? FSH and LH. And notice, we are using a 28-day or so cycle. Got it? And halfway in the middle, by day 14, is when we have the ovulation. Notice that we are also increasing our estrogen and progesterone. Alrighty? And your uterine lining is building up. 10-4? It's building up. By the time we get to the last day, if there is no baby, the estrogen and progesterone goes down because it's made by this guy. It goes down, and now FSH and LH kick back in to have you lose your lining. And why do we do that? Because we want to have a new functional layer every month, just in case a baby comes by to be implanted. We don't want them implanting in old tissue. We want them implanting in new tissue. And that's because that new tissue can then quickly grow and become a placenta. Okay? For the baby. So we can exchange waste and nutrients. Okay?
Say the name of the layer that is shed again.
Functionalis.
Functionalis.
Okay. Functionalis. And the one that's not shed is the basale. Okay? Yeah, we shed the functional layer.
Oh,
And
yeah,
we don't want to shed this layer if there is a baby.
Mm-hmm.
That's why we want the corpus luteum to stay a corpus luteum for 3 more months, telling your hypothalamus, don't let the anterior pituitary release FSH and LH, because I don't want to have another menstrual cycle. But I don't want to lose the baby that's growing in this. 10-4? Now, why is that only for 3 months?
Then the placenta takes over.
Then the placenta has been created by that time, and it takes over for the rest of the 9-month period. Got it? Super.
All right. Function of corpus luteum. I feel like we kind of already.
Yeah. It makes the
Estrogen
estrogen
and
progesterone. Yeah. To be the negative feedback, I don't know if they got that in here. To be the negative feedback, I might have passed it. Yeah, see? There was a picture in the male part too. Hypothalamus says, hey, anterior pituitary, gonadotropic releasing hormone, release FSH and LH to make sperm. Well, this is make LH and FSH to tell the egg to ovulate. Got it? But at the same time, we make estrogens, which negatively feed back and say, okay, pituitary, stop doing it. Okay, hypothalamus, stop doing it. Okay? And the same thing, the testosterone that we make feeds back and says, okay, I've made enough sperm for today. Block this. Got it? That's why it's called negative feedback. Okay? Because this is going to feed back to the origin and say, don't make no more. We've got enough. Cool beans? Okay.
All right. Next up is gonorrhea, syphilis, and
Okay.
chlamydia.
The diseases. And it's not going to be very difficult. I'm not going to show you pictures of it and stuff like that. What we need to know, okay, they call them STIs now instead of STDs, okay? Sexually transmitted infections. I don't know why it's not a good idea to say AIDS,
I
but
think
disease.
the
But gonorrhea is known as the clap, okay? And what is it? Bacterial, all right? There you go, okay? With a clap doll. Don't ask me why. You can buy that, okay? Plush clap doll. That's what it actually looks like, not this. It looks like a paramecium. But anyway, syphilis, again, a bacterial infection. That's what killed Al Capone, because it affects your brain, okay? And then chlamydia, which actually is the most common STD. And, okay, and sometimes you can't tell what's going on. That's another reason why it's so widespread. But those are all bacterial, which means they can be killed with
antibiotics.
antibiotics.
Yeah.
See, the other ones are viral, so they have to be dealt with in a different way, with an antiviral. Trouble is, being a virus, it's not alive, and so you really can't kill it. So there will always be viruses that will survive by hiding inside the somas, the bodies of nearby nerves. And then when the conditions are right, they pop back out. Got it? So I might have said this on the video. It's the gift that keeps on giving. Because once you've got this, that's what you're going to be giving to your other sexual partners. Okay? So you got to be careful. Sometimes you can see the sores like a cold sore. Other times you can't, but you still got it in you. Okay? And there it is, like a little coronavirus kind of thing. Okay? All right.
And then the last one, what triggers ovulation? I think
Yeah, we're triggering a surge in LH and FSH. Mostly the LH.
that's
Cool? All right.